Welcome everyone. It's my pleasure to have Anandhi, Iyappan, and Karoline Mauer from these are the metadata experts from the German Human Genome-Phenome Archive, who are going to show us how to ensure fair data practices and how we navigate. How do they navigate the data portals? Our first speaker is Caroline Mauer. She is a PhD student within GHGA. She is involved in standardization and harmonization of data and metadata, as well as developing a platform for COVID-19 data. So, Caroline, stage is all yours.
Thanks. Thank you for the introduction. Let me just share my slides. Yeah, welcome from my side to the webinar today. So Anandhi and I will talk about fair data and fair data portals and also how to ensure that your data is fair. And yeah, I will take over the first part and talk about fair biomedical data. And then Anandhi will take over with the second part about fair data portals. So before we talk about metadata, we first have to talk about data. And data in itself is just potential information. It can be, it's raw, it's unprocessed, and it can be just a sequence of characters. But since you don't really know what you're looking at, you first have to add context to it in order to understand it. Once you add context to data, it becomes information. It's now processed in human consumable data. Information can be analyzed and visualized, and it can also be measured, so you can use it to answer the first questions you might have. And once you apply information, it becomes knowledge. So in the knowledge stage, you also compare to your surroundings and try to put your knowledge into the greater perspective. And you can also ask how to use the knowledge to reach the goal why you collected the data in the first place. And once you do that, your knowledge becomes wisdom. So wisdom is applied knowledge, and now you ask, how can I use the data, information, and knowledge I have to achieve my goal? And in order to walk up this hierarchy from data to wisdom, you not only need the data, but you also need the context and the description of the data, which we know as metadata. Yeah, so this is a very abstract example. So I brought an example from my field of research, and I am working in omics medicine or biomedicine. And I hope you know what FASTQ files is, but if you don't, we can have another example at the end of my talk. Let's say you have a sequencing experiment, and in this case, it's a transcriptome experiment. So you want to study the expressed genes at a certain point of time in a human. And your first step would be to get the data, which are FASTQ files, and this is what FASTQ files look like. So as I said, it's raw, it's unprocessed. You know that there's something in there, but at this point, it's just a sequence of characters, and you have no clue what's going on. So the first thing you do is you extract the information. And for this, you already need the first metadata. You would know what organism you're looking at, which ensemble version you use to annotate your genes. And if you do that, you get normalized and annotated gene counts, which you can use for your first plots, for example, for quality control. And if you go one step further, you do your second analysis and you study the differentially expressed genes. And in order to do this, you need another set of metadata, maybe the patient groups or the disease you're looking at. And if you have done this, you get to the knowledge phase, and this would be the classical discussion part of any study or paper. You look at the information you have and ask, do my findings align with the literature? Is there anything that I found new, or where does my information close any gaps? And at this point, you are reliant on the information that is already in the vicinity of your study. And then in the knowledge phase, you can maybe identify possible treatments based on the genes you have studied. You can design follow-up studies or replication studies, and maybe in the end, design a drug to treat the disease you have been studying. And of course, for this, you would ask, how does my knowledge help me in achieving my goal of treating a specific disease? And as you can see from the boxes here, there is a lot of different types of metadata that you would need to get from data to wisdom. In general, there are 3 different kinds of metadata. We have administrative metadata, which is needed to manage data and data access. These could be access rights, creation times, physical and digital storage location, and also file formats. And the administrative metadata can also contain instructions on how to do long-time archival or version control. The second part is structural metadata, which is needed to store and retrieve data from a database and also necessary to understand the data structure. And here we have classes and entities, the cardinality between classes and entities, and also the relationships. And the third part is the descriptive metadata. I think this is the type of metadata that most researchers actually deal with. Here we have information about author and creator of data and metadata items. We have title or abstract, topics or categories that are covered by the data, and we have keywords. So if you have a clinical study, for example, what is noted down as keywords would be the clinical metadata. So for example, disease types, measurements, and so on. And this metadata type is necessary to find and understand data. If we would try to find adjectives for metadata in biomedicine, I always think that one of them is high dimensional, because metadata in biomedicine, there are a lot of different entities that you have to cover. We have individual samples, protocols, studies, designs, relationships, analyses, and many things more. It is highly valuable, not only in biomedicine, but in general, because without any data and metadata, there would be no research. It is relative, because one person's data is another person's metadata and vice versa. It is sensitive, because we deal with detailed clinical and social data. And you can ask whether sensitive data can even be fair. Just as a reminder, FAIR stands for Findable, Accessible, Interoperable, and Reusable. And it basically says that others should be able to find your data. They should know how they can access it. It should be readable for humans and machines and can be interacted with. And it should be well annotated and can be used for diverse use cases. The FAIR principles were published in 2016 as a guideline, which I find very important to highlight. And they stress the ability of machines to find and use the data, and thereby aiding a human to do the same. They were published due to the explosion of data that we not only have in the biomedical field, but everywhere, the increased international collaboration, and both of these cause extreme data management issues. And another thing to remind you of is that fair data always refers to a fair digital object. And a fair digital object consists of these 4 elements. We have a persistent identifier, we have the metadata and information about the provenance. So in my example of maybe a sequencing experiment, the provenance would be the protocols that were used to get the data, and the data then itself, like gene information. And this digital object is only fair if all of these components are fair. Why would you even want to be fair, although it's pretty difficult? So why would you want to share data? First, it's part of the scientific process, and it's necessary to reveal the validity of the findings. And it also enables others to answer questions that they might have based on your data. Second, you should lead by example and encourage others to share data by doing it yourself, and thereby everyone benefits from data availability. And lastly, you can say that fair data advances science and it builds trust. What happens when data is not shared can be seen in the reproducibility crisis. And here I brought a paper from the Reproducibility Project Cancer Biology, which was published in 2021. And they tried to replicate 193 experiments from highly cited papers in cancer biology that were published between 2010 and 2012. And the authors say that the authors of the studies say that the authors of the other studies either do not share their data at all, or they make it difficult to understand. Maybe it's on purpose, maybe not. And they find that among the studies that provided the sufficient information to be redone, the effect sizes were on average 85% smaller than the original findings. And positive results were half as likely to replicate successfully than null results. And in the figure on the right, you can see the number of experiments they designed and the number of experiments they completed, and also the barriers they faced. And I want to highlight here that all of the protocols needed clarification. So there was not a single experiment that could be done only with the methods section of a paper. In around 60%, the authors were kind enough to help to replicate the experiment, but shockingly enough, only in more than 50% of the cases, the data was not shared at all. And you can only speculate why the data was not shared. With regard to this, I found an editorial from the former editor-in-chief from Molecular Brain, and he writes about his experience as a role as editor-in-chief. And he said that over this time that he served this position, there were 41 manuscripts that where he requested additional raw data to support the results. Of these 41 manuscripts, 20 were immediately withdrawn from the authors, and the additional 20 that then supplied the raw data, of these, 19 were later on rejected. So of these 41 manuscripts where he had to ask for additional raw data, only one study in the end was published, and all of the others were rejected at different stages of the process because the raw data was never supplied or it was inefficient. And he hypothesizes that the raw data did not even exist in the first place. But even if the raw data was there, just pushing it online somewhere would not make it fair, because without standards, infrastructure, and policies, it would be unfindable because it would be unorganized. In addition, especially if we're dealing with health data, we have to find a trade-off between findable and accessible and secure data. On the one side, we have the fact that we need accessible data to foster biological discovery, and we need detailed metadata to make the data findable and reusable. On the other side, since we're dealing with sensitive data, we cannot disclose personal data to unauthorized persons, and we have to ensure the protection of sensitive data. And often this balance shifts towards a secure side for the reasons I've listed here. There are risks for individuals, and the risk of losing trust in the researcher or in the infrastructure if there's a data leak. Then often FAIR is misinterpreted, especially findable and accessible, with regards that people think only open data is accessible. The implementation of the FAIR principles is very difficult. There's the lack of infrastructure and standards, and of course we have legal obligations, like I mentioned with the GDPR. You should also care about the GDPR because there's growing awareness for the data privacy concerns and solutions. For example, the UK Biobank enforces a mandatory training if you want to use their resources. We have a shift from data sharing to data visiting, simply to keep the control over the data and to be sure that it's not replicated elsewhere. There are trusted research environments and secure processing environments, and also swarm learning that is developed in the house where I'm working. Then, of course, there are risks, not only for the data subject, but also for the people and the employers that are working with the sensitive data, and it's necessary to know about them. And again, knowing about data protection is necessary to establish trust. And here I want to highlight that GHGA is co-organizing or co-developing Assured, which is an NFDI cross-consortia training for safe and ethical use of sensitive, potentially disclosive data for the research community. And if you want to know more about it, yeah, feel free to reach out. And I think Vanessa might also be in the chat. So yeah, just a little advertisement block. So yeah, how can you make your data fair? And I often see it in grants and proposals that there is the stress that researchers should do something. And yes, researchers can do something, but they cannot do everything. So of course they can implement certain standards and make sure that their data is standardized and everything, that they choose data standards that are used in the field. But then there is a lot that depends on the infrastructure. For example, making data findable and accessible depends on the data portal and also on the data subjects you're working with. Things you can do is that you think about what your data can deliver and you can make a metadata wish list. You should definitely harmonize and standardize vocabulary, and if necessary, use a data model to structure your data. You should have a data dictionary so that after years of data collection and analysis, you know what you collected in the first place. You should have domain relevant standards. You should publish the methods, results, and limitations of the data. And you should definitely choose your data portal wisely. I have an example in the next slide. Things you just have to accept is that detailed annotation might not be possible, especially if you're working in the health field and you work with real world data, so primary care data that comes directly from the clinics. Of course, no one spends the time to go over your very detailed metadata wish list to collect every single point that you want to have. Then, especially if you're working in a very new and upcoming field, there might simply not be this a wide range of standards that have been accepted. And accessibility and findability might be limited by GDPR or ethics. Now I brought you a real life example from my life as a researcher. So recently I had to download a lot of data. So I went to a data portal and grabbed everything I can. And I had thousands of studies. And here I have a list maybe of 4 studies. And when I looked into the metadata, I was quite shocked because study ID and sample ID, all of them were standardized, which was fine. One of the columns was a sample source, which was fine enough, but then again, it was not standardized, which you can see because PBMC and peripheral blood mononuclear cells are the same thing, but they are written differently. And after that, everything was left to the submitter. So they just said, enter something in column 1 and enter something in column 2 until you are done. And here you can see that it's not even harmonized how to differentiate key and value pairs. And when I tried to tidy this up, I was left a bit puzzled. So these highlighted things were the points I was interested in. And I tried to come up with something that is harmonized. And the sample source was pretty easy. But when I looked, for example, at the sex, there was one study I could use. But for example, for study 3, I don't even know if patient gender is the same as the sex that was used in study 1. So this is one example why, although the researcher made their data very reusable, if the data portal is not doing it, then it's not reusable. And this shows that data portals need a definition of a minimal set of common and standardized properties. So again, the burden lies on the infrastructure and not only the researcher. Core fields that, at least in biomedicine, should be considered are age, sex, disease, case control, and sample tissue for the individual. On the technical side, we need information about a data type, instrument model, pipelines, software and software versions. And for the administrative data, study type data controller, maybe a place of collection, modalities, and an abstract or protocols. I also said you should or can use a metadata model. I think a data portal should definitely use a metadata model. This is a representation of captured data, and it allows to structure data to define relationships between entities, to describe these entities using properties, and to store and query data in the database. There are a lot of existing metadata solutions, and these are just the ones I have encountered and worked more closely with. So there are agnostic solutions that work regardless of the data type you have at hand, like research object crates, schema.org, linkml, JSON LD, RDF, DCAT, and DataCite, they more or less allow you to define the metadata model like you need it. Then there are metadata solutions that work extremely well with provenance data, which records the changes that were made to a file and also the pipeline that was run, like workflow hour creates or biocompute objects. And then we also have a lot of biomedical data models like ISATEP, bioschemas.org, which again are very broad. We have clinical models like OMOP-FIRE and PhenoPackets, and two German solutions, which are the core data set by the MII, the Medicine Informatics Initiative, and NFTI for Health. And I want to end with some pitfalls and things to consider when you're dealing with fair metadata. You should definitely align to standards and other schemas, because there are already a lot of community accepted standards that help you to verify your data. And maybe you all know the XKCD comics about having 13 standards, developing a new one. Now then you have 14 competing standards. So you should definitely only develop something new if you really have to. Then you should make use of ontologies and controlled vocabularies. If you define metadata and don't control what people can put in there, then you don't even need to bother in the first place to set everything up. Yeah, third, as I said before, accessible data is not equal to open data. To be fair compliant, you have to be accessible and not open, and vice versa, only because data is open and does not mean is accessible. And then lastly, if you're dealing with sensitive data, you should definitely know your GDPR and your data protection officer, because biomedical or human biomedical data is considered sensitive under the GDPR, and it requires special protection and processing. And lastly, I have some additional resources from GHGA, so feel free to check out our YouTube channel where we have many more webinars. And with that, I'm done with my part and I'm open for questions.
Thank you, Caroline, for a wonderful presentation. It was very informative, and I completely agree that we should all, yeah, I mean, encourage also each other to submit the data on the data portals in order to keep the transparency and reproducibility. Yes, so and like coming from publishing industry, I on daily basis I do see challenges encouraging authors and asking them to please do share your data. And yeah, so yeah, as Caroline said that she's will take now some questions before we move on with the presentation. So if anyone has question, please drop it in the Q&A box, or if you want, you can also ask your question live. Okay, maybe I can ask one question. So, as I said, that we do see on daily basis challenges to encourage authors and to ask that please do share your data. Once I experienced that there was a reader who wanted to access the data, but after publication, the data files were removed from the data portals. They did submit the data, deposit the data for the publication because it was necessary in order to have publication. But then, what do you suggest in such cases? For the data portals, do they receive some kind of notification or something that files have been received or something? I mean, is there any sort of control or something technically, or I don't know? I mean, because sometimes we don't know, because talking about paper mills and all this stuff, we don't know if these authors do exist or when they're submitting their data, and even if journal and publishers are trying to contact them, I mean, sometimes you might not receive any answers. Then what should we do with that?
Yeah, I think it depends on the data portal and also how they allow people to change the data after submission. So I think in some data portals, submitters are relatively free to do with the data whatever they wish, to change things afterwards, to add samples or to update things. And there, of course, the trust is that people don't abuse this power, right? But
Right.
what we are also discussing for GHGA is how we, on the one hand, enable submitters to update their data if they, for example, have additional samples or they entered the sex for one individual wrongly or stuff like that. But on the other side, make it transparent that a change to the original submission was done, where
Yeah.
the change was done, and why the change was done. And of
Right.
course, yeah, if the submitter just is able to withdraw the data after submission, I would, yeah, see the fault in the data portal. But sometimes these data portals handle such a large number of submissions that maybe they don't care about the black sheeps, and I don't know.
Right. Yeah. Yeah. Thank you very much. We have one more question. Please, would you like to ask yourself, or should I read it for on your behalf? Lucy, can you give access to? Yeah, thank you. Okay. Sorry. So I will read the question. So when it, so bet, so please excuse me not to pronounce your name correctly. Beatrice, right? Yeah. When it comes to fairness, some data will always be more fair than others. Is there a scale or organization that certifies fairness, and who is responsible for ensuring it?
Yeah, that's a that's a tough question.
Interesting.
Yeah, first of all, I would say that fair is also a process, and I find it very hard to measure it. I think there are like fair scorers or scoring matrices, but most of them rely on the person that wants to know how fair the data is to enter the information. So that might be a bit skewed. There are resources like fairsharing.org where you can, yeah, I don't know if you have to just register it or if someone looks over it, but maybe Anandhi knows better than I. Yeah, this is a resource where you can check if the thing you're looking at is actually fair. Who is responsible for ensuring it? Like I said, if you are a researcher that has data, then of course you are reliant on the infrastructure to also allow you to make the data fair for a wider audience. So I know that often this burden is pushed on the researcher themselves, but I don't think that is fair and that is the way to go. Yeah.
Okay. I hope that answered your question. Thank you for asking the question.
Yeah,
So
thanks.
if we do not have further questions, I would like to thank Caroline for the very wonderful presentation, and I would move on with our next speaker, who is Anandhi. Uh, she's uh, um, she's currently working as a data and meta standards officer at GHGA, where she's responsible for harmonization, standardization, and generation of data and metadata. So, Anandi, floor is yours.
Thank you so much, Manu, and please let me know if you can see my screen. So thanks again from my end for all of those who decided to come for the presentation today and also decided to stay for the second part of the presentation. As we already saw in the first part of the presentation, we learned about FAIR principles in general and what are the do's and don'ts as far as researchers are concerned and how they should approach. In this part of the presentation, we will be focusing more on the existing communities and the existing data portals, which relatively are consortiums and communities which support fair sharing, and also some data portals which implement fair sharing to some extent in their in storing data for their repositories. To start with, in biomedical research in general has a lot of data management requirements as part of, say, funding agencies or as part of policy making or requirements from the community itself. And this puts in a lot of liability on data producers and data consumers on how they produce their data and how they store their data. And having said that, I would like to directly move on to the reason why we need data portals. As I mentioned, these are also day-to-day requirements in today's day and age. Data portals in general are like websites or platforms that are designed to store, publish, and share data from different applications and also from different kinds of users. Open data portals in general are designed to allow researchers to be able to submit their data in an efficient manner, to make the data searchable in ways it can be searchable. We spoke about FAIR, so searching also comes with a lot of liability, but still a platform that would provide the option to search information, and at the same time to ensure transparency in the kind of data that is being stored and the data that is being used. The European Commission also came across a 7 or a 10 pointer on how data portals should be designed, or what should an open data portal contain or consist of. So that you can see in the figure, the data that in data portals should be discoverable, they should be measurable, they should be accessible, they should have strong metadata information backing up the data that is being generated. Usage of standards, organizing the data, documentation. Also, if there are any additional data that links to the existing primary data, and also ways in which that can promote the usage of that kind of data. So all of these principles are provided by various committees and organizations, but it also depends on the purpose of data portals, to what extent they can implement these pointers that have been provided. I'm having issues with changing my slide, sorry. Yeah, okay. As we saw that there is, first of all, open data portals. The next question that we need to ask is, why do we really need data portals, open data portals for that matter? The first aspect is to strengthen the transparency, as we already saw on the previous slide, to foster better research in general. Transparency improves trust between the producers and the consumers of data. As far as decision-making is concerned, decision-making as far as data is concerned, data in general has no value, right? But a data can create value only when it is being shared, used, analyzed. So if such data can be properly maintained and provided the necessary information, then it becomes very valuable, and therefore it also helps in precise decision-making as far as research is concerned. And of course, the need for data, the availability of data also helps us to develop newer services, newer tools and pipelines to analyze the data that comes in. And this, at the end of the day, it also helps in fostering collaborations, because data exchanges are very common phenomena that happens globally these days, and open data portals helps very much in fostering such collaborations. And on the right-hand side, what you generally see as far as biomedical research is concerned is that new data portals or open data portals also promote newer research ideas that are generally very efficient. And also it enables long-term availability and communication. And also, it helps in fostering not just research, but also advancements in tools and technologies. And also, when such data are properly published and they are provided in a standardized manner, it also adds a lot of value to the citation or the publication resources, where the data becomes valuable and reused. And also, there are stronger economic benefits because it promotes various projects, and that in turn generates a lot of revenue. So it does come with a lot of advantages. What we saw so far are about open data portals. Right now, I'm going to talk a bit more on the FAIR data portals. So what is the difference between open data portals and FAIR data portals in general? Data portals are definitely designed to store data, but as Karo also mentioned, data can always be nasty, dirty, not properly standardized. So there is always a growing demand for the kind of quality of data sets that can be produced and stored and also managed. It's not just about keeping them, storing them and managing them, but also coming up with stronger infrastructures where that data can be shared in a reliable and in a trustworthy manner, that there is some credibility to the data that is being produced and being submitted to a particular data portal. This clearly differentiates between generic open data portals and fair data portals. So on the right-hand side, what you see is pretty much like a structure or a representation of how a fair data portal should be designed. So it should come up with various functionalities, or it could come up with various functionalities like archiving facilities, information that are stored that can be retrieved, that can be analyzed, and also coming up with stronger support system from the data host and to the data consumers as well, where there can be a stronger interaction between the two entities, and also stronger legal formalities or legal bodies which control the data in a legal and in a responsible manner, in such a way that there is no breach that can be encountered while using the data or while submitting the data. So all of these can be additional entities to data portals to make the data more authenticated and also stored in a more safe and secure manner. Having mentioned about data portals and fair data portals, now we will be looking at the existing research communities which support and foster open science, open life science research, and also fair data principles or practice fair data principles. Elixir is one such organization which came into existence in the year 2013 as part of the European Infrastructure for Life Science Research. And this was mainly formed to ensure and enable that the different national centers as far as bioinformatics is concerned to be coordinated in a single coordinated infrastructure. So this is a platform which helps in bringing different data resources in one particular place. And it underpins also the management and safeguarding of the data that is generated by publicly funded research as well. The main mission of Elixir is to ensure that the data that comes or is part of their infrastructure is findable, analyzable, shareable. And also it consists of expertise and different experts coming from the community, which also acts as a platform to exchange expertise between communities and also between researchers. And also they have come up with stronger guidelines which the communities can practice to enable best standards practices as far as their research is concerned. Sorry. The Elixir is also not just functioning in one particular location. It is a distributed European research infrastructure. It consists of about 23 nodes and present over 250 organizations across different countries within Europe. And also, they have developed a very strong, trustable domain where repositories are created in such a way that they can be curated through the nodes. And many such data portals and data repositories are also working in tandem with Elixir, promoting global research and making sure that all the researchers from different countries benefit from the services provided by Elixir. Also, they ensure that they support data repositories by enabling or by fostering FAIR principles, like ensuring that the standards are being established, identifier services are being put in place for the services that are being developed, and also scalable curations are taken care of for the data, and also making use of standardized representation of their model or the data in the form of bioschemas, to name a few. The next community that supports fairness or promotes fair data is Research Data Alliance. So Research Data Alliance was also founded in the year 2013 by the European Union and the American Food and Drug Association. It is also an international organization which has a strong focus on infrastructure and community development that also reduces barriers in such a way that data can be exchanged and shared across the globe. This particular community is quite unique for their focus groups that they came up with, which in 2013 was very new. Today, we might be very much aware of community and focus groups, but they were the pioneers in setting up focus groups where researchers from communities can come and discuss specific topics, which also helped in advancing the concepts of reproducibility, how data should be preserved, best practices that should be practiced in different domains, How can a study curriculum be developed in these areas? How can data be cited? How can metadata be designed? So they were the first organization which actually took a step in coming up with these focus groups, which also enabled research to advance to a great extent. The third aspect is fair sharing. Caro already briefly mentioned about fair sharing. This is also a community which helps in ensuring to support curated, informative, and educational research on any kind of data and metadata standard, which can be implemented on databases and also associating themselves in coming up with data policies for fairness. They establish and promote open standards, ensuring that how should reporting guidelines be done, the different terminologies, exchange formats, ontologies, taxonomies, to name a few. So far, fair sharing has developed up to 1,757 standards across 2,000 databases, and they have also come up with various policies across the wider scientific community. Also, the question from Beatrice, maybe I would use the space to answer here. So they have like a metric system where if you can submit data to them, they will be able to come up with a score and to tell how fair the data is. And also rating fairness is very subjective because it depends on what we are aiming to achieve in that particular project. So even fair sharing has the tool which can be customized and can be evaluated for the fairness. And it is very subjective to the data that is being submitted to them. If we want to discuss this, we can also discuss about this at the end of the presentation. The last community I want to address is the Global Alliance for Genomics and Health, which is a relatively newer consortium that was put together to aim to accelerate the progress of genomic research and human health by developing standards and also by coming up with specific policies in genomic research. Their main objective, they also have various community groups and which are actively taking part in discussions on how to improve the metadata of genomic medicine, how to improve the infrastructure, how can community outreach be enhanced. And their main objective is also to enable data sharing, interoperability, how to improve privacy and security of human genomics data. What are the ethical and legal considerations that needs to be taken care of while handling human genetic data, and also how can we innovate from the current state of genomic research to the future. Now, having seen the different communities that support fair sharing, now we will also take a look at various data portals from the time as early as 2010 to the recent ones. The first database that was was used very predominantly by researchers is NCBI Geo. The Gene Expression Omnibus obviously is a database repository that is used highly for analyzing or knowing about data sets related to gene expression. Chip-based experiments, assay-based experimental data were submitted to the NCBI Geo. It was also one of the most popular publicly available archive for storing and using gene expression data. The data portal looked like this. So it was possible for people to search using keywords, as you can see in the bar, hepatocellular carcinoma. And with that search function, it also had possibilities of filtering the information for getting the kind of data sets that you might need. You would be able to filter based on the study type, organism, author, and you will be able to receive the corresponding data set. And it also had the possibility to come up with analytical tools like gene expression analysis, differential gene expression analysis in their own data portal. And also it was possible to visualize the results of the gene expression analysis that were performed on the data portal. This was one of the first data portal that provided these interactive platform for researchers to enhance their research. The next database is Genomic Data Commons, shortly called as GDC. This is also a platform that allowed researchers to search, analyze, and download cancer genomic data. The data portal had a very interactive web interface, which had information about the anatomical information about a human, and also the different organs and tissues from which the data sets were generated, the kind of genetic information, mutation-based information, and that also consists of data coming from across 83 projects as well. This particular database came with a cohort builder, which helped in also filtering the data sets based on features. As we saw in NCBI Geo, which was much more simpler, this consists of much more, it has a wider or a diverse range of filters or fields based on which a person can choose the data set that they might need. This has detailed information on the project with also options to choose from it, so it helps in very specific customization of data sets as well. The third data portal is the CBio data portal or CBio portal, which is a platform for exploratory as well as interactive visualization and analysis of large-scale genomic data sets. This basically consists of information from literature as well as from curated databases or repositories containing information related to therapies. And it also provided a platform to analyze the network-based data along with the literature data, which helped also in bringing research data and literature data together and to search information. It was also possible to upload clinical and proteomic or any other omic data to this particular data portal as well. Now that we have seen about data portals and data communities, now we can also see what are the various ways in which fairness in data portals can be assessed. As we constantly have been speaking in this presentation, it's extremely important to know about not just the data quality, but also what kind of implications we can have when the data quality is quite poor. So it is quite obvious that with the amount of research that's going on these days, there's also a lot of data that is being produced. So in order to do quick research, it can also so happen that many data portals upload data in a way that it is very messy and not so accurate. So the obvious outcome of that would be garbage into the data portal will obviously be garbage out of the data portal. So this is basically making us going to go in circles, and it's not going to help us progress in the research that we are conducting. So it is very important to understand what are the implications of having poor quality. Only that will help us to see what are the ways in which we can improve the quality of data. So a poor quality of data will obviously cause inaccuracy. It'll lead to a lot of redundant information, and it can lead to a lot of incompleteness of the data. And also it's highly expensive because the research that was done can never be reused by another person, because the research might have been a great research, but the quality or the output or the outcome in which it was stored was not done in a way that somebody can make use of it. So in order to assess the fairness, there are certain metrics or indicators which can be adapted to evaluate the quality of data sets. There are various organizations which have come up with templates. This is one of the most popular templates that exist in order to consider a metric or a fair metric. So as far as data is concerned, we can ensure that the data is fair if we can ensure that this information can be captured in the particular database, like an ID, a metric name, to which principle does the metric apply. Here it means if it's the F or the A or the I or the R, what is being measured and why are we measuring it and how do we measure it. If these kind of questions can be captured as part of data, it is already a decent metric which says something about the quality of the data. As I mentioned to you about the different communities earlier, RDA metric, the Research Data Alliance, have also come up with a similar metric, which ensures that the metric that is developed is useful, important, as well as essential. They have come up with fields or like indicators which mark each field with these three labels, whether it was useful, whether it's important or essential. These data portals can adopt to capture information from the users and also ensure that the data that is being submitted is complete and does not remain incomplete. Also, fair sharing has come up with various ways of analyzing the fairness of data. They have come up with fair metrics like maturity indicators such as fair enough and fair shake. RDA fair data maturity model, which has a fair evaluator, which evaluates using certain functionalities how good your data is, how fair your data is. And also there's something called fair sphere data object, which also has its own assessment metric, which helps to check the fairness of your data. As I mentioned to you in the beginning, fairness is very subjective and it depends on what we want to check for the data, what is it that we are looking for in a data. Accordingly, there are metrics to evaluate the data as well. So as far as data quality metrics are concerned, it is always based on these 5 modules that you can see. One is completeness, second is correctness, third is conformity, fourth is consistency, and fifth is coherence. All the fair metrics are majorly based on these 5 modules. And these metrics basically helps us to identify what needs to be there, but they also ensure that they don't tell what is necessary and what is also sufficient. And evaluation of these fairness also reflects on which type of a digital resource addresses these fair principles. As I mentioned, it's very subjective. And fair metrics are obviously very good enhancers of the data because it adds a certain quality and reliability to the data that is being captured. So, and this obviously is going to enhance better reuse of data, better discovery of data, but also it is going to promote the community interoperability as well. Now I'm just going to show you a small example of how a scoring metric can be used as an indicator. So as far as indicator is concerned, we can rank them according to these 5 modules as I mentioned. These can be called as KPIs. These are indicators and also metrics that can be used to evaluate the fairness of the data. So whenever there is a data, even if it's not a data portal, we can always ensure that we take these measures when we have a data. We can always first check for completeness, ensuring that all the fields are captured in the data, in a spreadsheet that we are capturing information. Correctness of the data, ensuring all the information we are submitting is correct with no spelling errors, with no inconsistencies whatsoever. And ensuring that all the data elements conform to a defined standard. For example, if you're using units, using the right units and not using the wrong ones. Ensuring that the information that is being filled in the entire data sheet, that it's being consistent. You're not writing male in one place, M in a different place. And also ensuring that all the information that is being submitted is coherent across all the fields as well. So these are some smaller measures one can take to ensure that the data is properly standardized. This is one step that we can do as local data users, and also databases can adapt these when they are providing those services. Here I just wanted to show a small example of how a scoring metric is, or how fairness can be scored. So the first one that you see is for completeness. You have 2 rows. One row shows you that the information that has been filled is correctly done. And in the second row, you can see that the information for sex and the treatment is not mentioned. If you look at the correctness, obviously the information in the first row is incorrect as far as sex is concerned, and also the tumor location is not correctly mentioned in the second example. So this can be evaluated by a score like this. So if you have like 10 rows, then 5 out of 10 is correct, then there will be a percentage for that. So this way, a percentage of the data set can be evaluated for completeness, for correctness, for coherence, and the other 3 other things I mentioned to you in the previous slide. So like this, fair metrics can be designed. And like I said, this can also be made subjective according to the data that we have in hand. Having mentioned about fair metrics, looked at communities and data portals, now we can just look at how submission to a data portal looks like. One very popular example, which I'm very familiar with and I quite like, is the ENA data portal. So they have a web-in data, like a portal where you can actually log in and go and submit data into their data portal. And this submission into ENA is controlled. They have a data controller, which the data is basically controlled. They have good legal measures there to protect the data that is coming in. And also the functionalities of ENA is to ensure that you can submit your data in the form of an XML file or also in the form of a spreadsheet. And it is quite an interactive platform for users to submit their data. So as I mentioned, they have a proper login where you can register and log into their data portal. And once you log in, you also have various checklists that they have provided in their data portal, which you can use for choosing your type of submission. For example, if you have a human gut based submission to do, then you can check for that particular guideline. And if it's going to be a different organ, you can choose the respective guideline as well. This is just an example. They have a lot of checklists in place for customizing it according to the data that you have. And also they have a very nicely curated spreadsheets, which is very useful for submitters to submit data to them. It has very clear explanation of what information is needed and what is the description of that particular field, which helps and enables the users to submit data to them in a very efficient manner. So they also mention what is important, what is mandatory, and what is optional information that you need to provide, which also helps the users to fill in the information. So this is one example of how an experiment information is captured and how sample information is captured as well. Having given a lot of broad talk about fairness, fair data portal, I think it's also very important to mention about the project that Karo and me are working on. It is the GHGA project and why GHGA really matters.
Sorry, Anandhi, to interrupt. I think we are running out of the time.
I'm almost done.
Yeah, okay. Thank you.
So GHGA is the German Human Genome-Phenome Archive, the project that is focusing on ensuring that we provide a secure omics infrastructure for storing human genomics data in a safe and responsible manner. And also we ensure that the data is highly protected, and we also ensure that we will not cause any kind of, preventing any kind of misuse of the data. This project is phased across 4 stages. We are currently in the archive phase, and we are moving towards the SPE as well as the Atlas phase in the near future. GHGA mainly matters for us because the model or the model that we are building is aligned with national and international standards. We are also trying to balance it between the data reuse as well as protecting the data privacy. And we are able to customize the model for different modalities. And also we are able to quantitative and qualitative metadata submission with varying degrees of freedom for the submitters. We can customize submissions as well. And we have various webinars on metadata. You can also check out on our YouTube channel and watch for these webinars on metadata that Karo and I have given. And also to just summarize that metrics were very, very important and essential for improving the quality of data submission, and community initiatives are very much important to foster research and enhance the usage of FAIR principles across researchers. With this, I would like to thank all of you for staying until the end, and happy to take any questions.
Thank you, Anandhi, for a wonderful presentation. And thank you for giving us an overview of different communities and talking about fairness of the data portals. I have one question for you. So how data portals or GHGA can help submitter, especially coming from the non-bioinformatics background or the first-time user to submit or deposit their data, especially like, for example, sophisticated data when they need to anonymize the data in order to keep it clean and storing the data?
So thank you so much for the question. In GHGA, I didn't have a lot of time to go in detail about it, but we have a strong, first of all, we have a good help desk management system, which helps you to collect the requirements. And we have like data stewards who will be interacting with the data submitters to understand the kind of data that they have, and to also understand and gauge where they come from and what kind of data they have. And also the data model is designed in such a way that it can be customized for different use cases and different modalities. As I mentioned, our metadata model is structured in such a way that it can cater to different types of data sets. And also it gives the user the freedom to be able to provide as much information that they can provide, But at the same time, there are also mandatory information that we expect from the users as minimum information that is needed to complete to enable the completeness of information. But because we have data stewards who are involved with the submitters, it is also possible for us to gain more information from them and to ensure that we have highly curated data that goes into the data portal. So this is how GHGA supports the fairness, and also the like I said, the help desk is also a very crucial interactive medium which helps the user
Right.
and the data portal to have stronger interaction and improves the transparency of what we have and what they want to submit with us.
Perfect. Thank you very much. Do we have more question in the chat box or in the QA? All right. In that case, I would like to thank our speakers, Anandhi and Karoline. Thank you very much for your time, and thank you for giving us this informative presentation. And I would also like to thank our audiences for joining us today with us. Thank you for attending. So yeah, so happy Valentine's Day and have a nice rest of the evening.